Hello everyone, I'm Sofia Arcelia and I will be discussing the first part of our group's report. So first, let's identify the parts of the intrapulmonary bronchial tree. As you can see, there's a hyaline cartilage, interlacing bundles of smooth muscles, submucosal glands, and a definite connective tissue adventitia. In successively smaller bronchi, the mucosal epithelium changes from pseudostratified ciliated columnar type with goblet cells. Next, we will describe the changes in the lining epithelium in the parts of the intrapulmonary bronchial tree. So the conducting piece of the respiratory system consists of the nasal cavity, trachea, bronchi, and bronchioles. The luminal surfaces of this entire portion has a lining of ciliated pseudostratified columnar epithelium with goblet cells. Their role is to secrete mucus that serves as the first line of defense against incoming environmental pathogens. Cilia move the mucus-bound particulate up and away from expulsion from the body. The various types and abundance of cells are dependent on which region of the airway they are in. In the most proximal airway, hyaline cartilage ring supports the larger respiratory passages, namely the trachea and bronchi, to facilitate the passage of air. Three major cell types are found in this region: ciliated, non-ciliated secretory cells, and basal cells. As the degree of branching within the airway tree continues, the epithelium gradually changes from pseudostratified to simple cuboidal, and the predominant cells become non-ciliated cells or Clara cells. Now let's compare the sizes, presence or absence of cartilage, muscular walls, and glands of the bronchial segments. Bronchioles are thin-walled airways lined by a variably ciliated or non-ciliated columnar epithelium. Bronchioles lack cartilage and glands which, along with their smaller size, distinguish them histologically from bronchi. Right main bronchus is wider, shorter, and more vertical, while the left main bronchus is smaller in size, but it is longer in length. The left main bronchus has two lobal bronchi, while the right has three. The primary bronchi has cartilage and the mucous membrane that are similar to those found in the trachea. As the branching continues through the bronchial segments, the amount of hyaline cartilage in the walls decreases until it's absent. As the amount of cartilage decreases, the amount of smooth muscle increases. In addition, hyaline cartilage forms an incomplete ring in the bronchi that gives them the characteristic D-shaped appearance in the larger bronchi. The bronchial wall is made up of mucosa, lamina propria, smooth muscles, and submucosa with interspersed cartilage. Bronchioles are made up of smooth muscle layers to facilitate bronchodilation and bronchoconstriction. The epithelial cells that line the bronchial tree are ciliated columnar cells, which are tightly packed and coupled by gap junctions. Lamina propria of bronchi also contain moderate numbers of mixed glands or bronchial glands. The bronchial mucosa is made of pseudostratified ciliated columnar epithelium with goblet cells and basal cells. Goblet cells are devoid of apical cilia and are responsible for secretion of mucin, and the basal cells serve as a reserve population by maintaining individual cellular placement in the epithelium. Within the bronchial submucosa and submucosal glands, these glands are composed of a mixture with serous and mucinous cells, similar to salivary gland tissue. The secretions are emptied into ducts and then on the bronchial mucosa. The pulmonary alveoli are sites where oxygen and carbon dioxide exchange occur during the process of inhalation and exhalation. Their lining cells are ciliated pseudostratified columnar epithelium with goblet cells, which is the same with the other respiratory passageways from the nasal cavity to the bronchi.
The difference of dust cells and Clara cells. Dust cells is an alveolar macrophage. It is a type of macrophage found in the pulmonary alveoli near the pneumocytes but separated from the wall. It is also a large phagocytic cell that moves about in the lumens of the alveoli. Dust cells are also known as the heart failure cells. It functions as a primary phagocyte of the innate immune system, clearing the air spaces of infectious, toxic, or allergic particles that have evaded the mechanical defenses of the respiratory tract, such as the nasal passages, the glottis, and the mucociliary transport system. On the other hand, the Clara cells are non-ciliated secretory epithelial cells lining the pulmonary airways. Its structure is tall, dome-shaped epithelial cells. These cells may also be found in the bronchi and the trachea. They represent the major secretory cell of the bronchular epithelium, where they contribute non-mucinous secretory proteins to the extracellular lining fluid. They also represent the principal site of xenobiotic metabolism within the lung, and they also represent an abundant progenitor cell pool for maintenance of the epithelium in the steady state and for regeneration of the injured bronchular epithelium. The structures forming the blood-alveolar air barrier is the epithelium and capillary endothelium with their adherent basement membrane and epithelial cell cytoplasm. It is formed by the type 1 pneumocytes of the alveolar wall, the endothelial cells of the capillaries, and the basement membrane between the two cells. The barrier is permeable to molecular oxygen, carbon dioxide, carbon monoxide, and many other gases. Pulmonary surfactant is essential for life as it lines the alveoli to lower surface tension, as a result preventing atelectasis during breathing. It is a mixture of lipids and proteins which is secreted by the epithelial type 2 cells into the alveolar space. Its main function is to reduce the surface tension at the air or liquid interface in the lung. This is achieved by forming a surface film that consists of a monolayer which is highly enriched in dipalmitoyl phosphatidylcholine and bilayer lipid or protein structures closely attached to it. Interstitium of the alveolar septa. The alveolar septum or wall consists of three components: epithelium, which lines the alveolus or the airspace, interstitium, and capillary endothelium. Gas exchange occurs in the alveoli across the thin epithelial lining and adjacent endothelium. Tight junctions are present between epithelial cells. Epithelial cells lie on a continuous basement membrane, as do endothelial cells. In many places, the basement membrane of epithelial cells and endothelial cells is fused, forming an extremely thin air-blood barrier. In other areas, the cells are separated by interstitium that consists of scant connective and elastic tissue and resident interstitial cells, macrophages, lymphocytes, plasma cells, and mast cells. And that sums up our report for today. Thank you for listening.